we can go ahead and get started. So thanks everyone for coming. It's my great pleasure to welcome you all to the CCBBI seminar series, and to kick us off, we're going to hear from Dr. Olivia Viessmann today. So Dr. Viessmann is a research fellow in radiology at the Harvard Medical School and Massachusetts General Hospital. Dr. Viessmann did her physics undergrad and master's degree in Germany and the UK, and she received her PhD in MR physics from the University of Oxford. She then joined MGH and Harvard as a research fellow, where she is now. So Dr. Viessmann is working on improving fMRI specificity by looking at the effects of vasculature on fMRI signals. She scans at max resolution to investigate fMRI signals as a function of head orientation. And she'll be talking about that today, as well as how to interpret fMRI activity in white matter. So please join me in giving Dr. Viessmann a warm welcome.
Yeah, thank you for this wonderful introduction. I'm very happy to give this virtual talk on V0 orientation effects today, which has been my main research focus over the last 3 years. My main interest is fMRI methods, in particular at high field and high resolution, to derive the most specific results in the individual brain. And this work will be a little educational session on the roots and the role of the vasculature on fMRI signals, but also in a broader sense on how brain anatomy and brain functional signals might be coupled. Let's see if I can, here we go. Okay, I would like to start this presentation with some teaser slides and foreshadow a little bit what results we will discuss today. So this is one of my favorite works from Bruce Fischel that nicely demonstrates a link between anatomical and functional properties of the brain. So this is Bruce's early work on the predictability of Brodmann areas from cortical folding patterns. And you can see that in the primary early developing areas, like the somatomotor area here, those areas have a high predictability of Brodmann areas, and higher order folds like here, they're not very good in allowing us to predict the Brodmann areas. And that was a nice demonstration of anatomic functional coupling early on. This is a result I will present in more detail later. So in this talk, we will visit cortical orientation, induced biases, and functional MRI signals. So here you can see an atlas of average brain orientation in the MRI scanner. And to the right, I'm showing how areas that are more parallel, like red, have a lower temporal SNR, and areas that are more perpendicular are those that have higher tSNR. For those that are not familiar with tSNR, it is somewhat a measure of signal to noise and kind of the currency of fMRI. And after this, we will leave the gray matter and take an excursion into the white matter, and I will show work that links vascular and fiber anatomy and show how both signals in the white matter might be related to specific vascularized fiber tracts. So this is just to give you a little bit of a little trailer of what we will be looking at. So here is the outline of today's talk. We will start with some theoretical background for you to understand the MR physics of how tissue orientation impacts MRI signals. And then I will show a couple of literature examples, and then I will present our findings in resting state fMRI in the cortical gray matter and show how orientation biases those signals. I will also discuss this at the group level. I think there are a lot of neuroscientists and maybe neurologists in the audience. And as you may know, brain anatomy and development varies non-uniformly across subjects. So anatomy-induced biases will also do that on the group level. And yeah, then we will dive into the white matter results, and I'll finish off with a little bit of a take-home message and a bigger picture on this work. So let's start with a little refresher on susceptibility-induced contrast in MRI. So MR contrast arises from the sample susceptibility, that is, the ability to get magnetized and distort the B0 field line in its local surrounding. So basically to act as a magnet itself. So in tissue, this is the content of iron, proteins, deoxyhemoglobin, myelin, and others. And the field line distortion of a sample depends on its relative orientation to the main magnetic field. So here is an example of a cylinder, which is often used as a toy model for blood vessels, but also for fibers. And this cylinder is changing its orientation to the B0, and field offsets are plotted through the cross-section on the right. And you can see how when the cylinder is parallel, the extravascular field offsets are minimized, and when it's perpendicular, they are maximized. So the field offset produces a gradient of precession frequencies that dephase the spins in the surrounding, and that is the signal loss we observe in fMRI when we sample a voxel that contains deoxygenated blood vessels. And there's an intra and an extravascular component. Both carry this orientation dependence, but of opposite sign. One goes with cosine squared and the other one with the sine squared. So this is roughly the root of orientation effects. And now I want to show you some examples from the literature where this has been observed in the tissue. So contrast can arise from the tissue's architecture, and that can arise on the macro scale, like large blood vessels and bigger fibers, but also down to the micro scale. So there are many examples in the literature where this has been demonstrated. This is a 70s study by Lee and colleagues. It's a white matter study, and they rotated an ex-vivo brain sample containing the corpus callosum in the scanner. And you can see here how the relaxation rate shows a strong orientation dependence with the rotation of this sample. And it is thought that this is mainly due to the myelin and not due to iron. So it's the fibers in the corpus callosum. And remember, the corpus callosum is a very unidirectional fiber. This is another study by Denk and colleagues who combined diffusion tensor imaging and multi-echo susceptibility weighted imaging to investigate the relationship between white matter fiber orientation and gradient echo phase and magnitude. And they really nicely show how the R2 star relaxation curve is depending on the fiber orientation to the main magnetic field. To also show an example where there is no orientation effect in parts of the tissue, this is a study of a rat brain. And here they rotated this rat brain and they observe, they sample here in the white matter, they sample in the gray matter and in the cortical gray matter here and in the deep gray matter nuclei. And they find orientation dependencies in the white matter, in the cortical gray matter, but nothing in the deep gray matter. So this speaks for evidence that there is no obvious tissue symmetry axis in the deep gray matter. And to show an example of microstructure-induced orientation effects, I would like to point out this study by Julian Cohen-Adat, who mapped R2 star changes with B0 orientation and fractional anisotropy from diffusion tensor imaging. So here you can see that the motor cortex has a high FA and a high R2 star change with B0 orientation change. So this association between FA and R2 star orientation dependence is interpreted as a higher structural coherence of myelinated fibers in this area. So now I want to dive into orientation effects in bold fMRI in the gray matter. Oh, yeah. So this is again from the literature, and the examples we looked at before, those were, the cylinder toy model, but also fiber structures. Both of them are geometrically less complex than the cortical vasculature, which is a hierarchical tree structure, and it's very complex. So here you can see a vascular anatomic network of a mouse cortical voxel. This is a reconstruction from optical imaging, and you can see there are large so-called peel vessels on the top, and then you have a kind of pseudo-random capillary network in between. And the first to simulate realistic field offset patterns from these van models was actually Louis Gagnon. And this is a really powerful tool to predict or bold fMRI signals from first principles using really realistic vasculature. And my colleague Avery Berman, he did a great job here. He did those simulations with this particular van, and he took that voxel and rotated it with respect to the main magnetic field axis. And you can see how for different rotations, these field offsets are really changing. And one thing that I would also like you to remember from this slide is that it's really these big fat peel surface vessels that have a really big extravascular field offset that goes way beyond them. So if you have a voxel that samples from this area, although it might not be touching those big large surface vessels, it will still be influenced by the extravascular field offset. So the orientation dependence of susceptibility effects, and more particularly and specifically in gradient echo-based fMRI, is really not news. You can already find notes on that in the very early Ogawa papers on the bold effect. However, for a long time, vascular orientation effects have been neglected, as the vasculature was thought to be random or voxels too large, and kind of saying, okay, our voxels are so large, we will get so many different orientations in there, we don't have to bother. But as you saw on the previous slide, the vasculature, first of all, is not random at all. And with higher voxel resolution, those effects might impart orientation signatures into the signal. So before I showed you this mouse van, here's a representation of human cortical vascular hierarchy. So this is actually from monkey. We have these big fat peel vessels on the surface. Recall those guys are dominating the extravascular field offsets. We have diving arteries and ascending veins and a capillary network that is said to be pseudo-random. Although I would like to point out here that there's more and more evidence also evolving that capillary networks also have an orientation preference with respect to the cortical surface. So a decade ago, we would sample 5mm isotrophic voxels. Now I think 2mm ISO is standard, and the high resolution state of the art is definitely pushing beyond the millimeter. So the first experimental evidence of cortical orientation effects linked to the vasculature were done by Louis Gagnon, who impressively predicted those effects from first principles using those van models, and then experimentally measured a 30% orientation dependence of the percentage bold signal change in a hypercapnia task. So for those that are not familiar with hypercapnia, it's kind of like the strongest vascular response that you can induce in the brain. Hypercapnia is just a fancy word for breath hold. And it induces really like a global brain change in blood volume, blood flow, and oxygenation. And this is the nice experimental curve that he showed, that with sampling at different locations with different orientation, he saw this change in percentage bold with this hypercapnia task. And more recently, Viessmann from Klaus Scheffler's lab in Tübingen took the simulation models a step further and showed that these orientation signatures are highly dependent on the depth where you sample your voxel from. So here you can see orientation curves for different depth of this van model. So there was clearly a lot of evidence for the existence of cortical orientation effects in bold fMRI and their relation to the cortical vascular anatomy. But as I said, Louis Gagnon's results were done with this very strong vascular challenge that is far beyond the vascular hemodynamic response that occurs alongside neuronal activity. So I ventured out to test if these effects impact modern day and state-of-the-art high-resolution gradient echo bold fMRI time series. So how do we do this in practice? We know that the orientation effect arises from the vasculature, predominantly from the peel surface vessels, but these are really hard to measure with MR angiography. So if you do a standard time of flight, you will not capture these peel surface vessels because the flow of them, although they're so big, the flow in them is like very slow. The big peel vessels are always perpendicular to the cortical surface normal vector. So we can rewrite the orientation dependence of the intra and extravascular bold changes as a function of the orientation of the cortical surface normal instead, assuming that effects are dominated by these big vessels. And I would also like to point out that the extravascular bold component is really dominant over the intravascular above 4 Tesla and is almost more than 90% at 7T. In practice, we use cortical surface reconstructions from anatomical T1 weighted scans. So here we use free surface reconstructions. We also calculate surfaces at multiple depths of the cortex to perform depth-dependent analysis and to really validate what we saw in the simulations. For each vertex of the surface mesh, we calculate the local surface normal vector and its angle to the main magnetic field direction, B0, and you can see this here in this color overlay to the right. The cortex smoothly varies orientation to B0 with cortical folding. So here I overlay the electron microscopy reconstruction of cortex on top, so you can see how all these peel vessels are perpendicular when the cortical surface normal is parallel to V0, so in this case here. And when the cortical surface normal is perpendicular to V0, then these peel vessels, they could have any orientation. So they could be parallel, they could be perpendicular. So in this configuration, the extravascular field offset is minimized. In this one, it's maximized because all the peels are perpendicular. So here are the specs of the resting state data that I'm about to present. We acquire data at 7T and at 3T. So the 7T data is 1.1 millimeter isotropic, total acceleration of 12. And the 3T data is 2 millimeter with total acceleration of 4. And we combine the orientation information from the anatomical scans with the fMRI data by assigning each voxel an orientation angle of the closest vertex to its center of mass. We then calculate the coefficient of variation, which is the ratio of the fMRI voxel's EPI time series standard deviation over its temporal mean as a proxy for the resting state fluctuation amplitude. So here are the results for the 7T data. So here's a plot of the coefficient of variation sampled at the peel surface, and you can see a voxel mass to the right. The yellow voxels are the ones that contribute to the plot on the left. And you can see that we really sample only at the peel surface here. So that's kind of like the worst case, the most impacted place we could sample from. And yeah, you can see that the bias here at the surface is 70%. And I would like to remind you, this is resting state data. There is no task involved. So what Louis showed in hypercapnia at 3T with like 30% bias presents as up to a 70% bias in resting state at high resolution and high field. Now I'm gonna slide through our depth analysis. So now we are moving, you can see it on the right in the depth mask, we are moving a little bit away from the peels, and you can see how this orientation effect is slowly like melting away. So now we're kind of at almost mid-depth. You can see these yellow voxels are really not touching the peel surface anymore, but we can still see this orientation dependence that arises from the peels. And this really speaks for the strong influence of the extravascular field offset that radiates far beyond the peels. Or another way to interpret this as well is that there might be a preferred orientation of the capillary network that runs parallel to the cortical surface as well. So if we go further down, this effect really doesn't go to zero, even though really none of those voxels is touching the peel surface. And at this point, we were expecting this effect to really melt down towards the white surface, and it's going down. Then what happened? It went back up. And we were a little bit confused what was happening here. And you can see here, we really took care that these yellow voxels, our mask, are not accidentally sampling from the opposite bank. So we're not kind of going back into the cortex on the other side. These are really voxels that have their center in the white matter. And we went back to some classical literature. We suspect that this might be an orientation signature of so-called principal vessels. Principal vessel is a term that was used by Duvernoy, who is famous for his study of the cerebral vasculature and who pointed out veins and arteries that dive perpendicular through the cortex and bend off in a perpendicular manner at the gray-white interface and kind of like run along the banks. And that kind of makes sense from a geometrical point of view. But that's definitely something that we want to investigate further, because that's kind of cool. So I won't show the 3T results in detail, but we were also able to detect the orientation bias in the 2 millimeter 3T resting state data to a lower degree though. So at 3T, the maximum bias was about 10%. So by maximum bias, I really mean that difference between a voxel that is sampling a piece of cortex that is perpendicular to B0 and a piece of cortex that has the surface parallel to B0. And here you can see this, the black plot, that's the 70 maximum bias as a function of depth. So it's like the worst, like it's 70% here at the gray matter CSF, drops off through the gray matter, never really goes back to 0, and then starts to go up again in the white matter. And the blue plot, that's the 3T data. So I also want to highlight that these effects are highly likely to depend on voxel resolution, but also on the field strength, because the field strength is driving this ratio of intra and extravascular field offsets. And yeah, interesting things happen at the white matter. That's definitely a space to watch. So all that we looked at so far were results that tell you something about the orientation-induced bias across the cortex of a single subject. And now I want to discuss this topic a little bit from a group level point of view, because we know that anatomy varies differently across the cortex, which is mainly due to evolutionary and postnatal development. So anatomy here, cortical orientation influences the fMRI signal, we just discussed that, and anatomy varies across the brain, but also across brains. So to the left, we can see the cortical expansion during postnatal development. And the lateral temporal lobe here is expanding to a much higher degree than the medial lateral occipital cortex. And the anatomical variation will be higher in this guy. And to the right, we can see how this is linked to also evolutionary development. The patterns of evolutionary and postnatal brain expansion are very similar. The early developed regions are those that are more similar across our brains. And the early regions, also called primary regions, this is something like somatomotor, and higher order folds, also often referred to as association cortex, are those that are really more variable in anatomy across us. And here I'm coming back to what I showed in the first slide. That was nicely really demonstrated by Bruce Fischel and his paper on predictability of Brodmann areas from cortical folding patterns. Higher order folds are more complex and have lower predictability than primary folds that are less complex. So I asked myself the question, what is the average brain orientation and how similar is it across brains? To do that, we used a subset of 110 HCP subjects to calculate an atlas of average orientation of the brain and orientation variability across us. And so here is the average orientation across those 110 subjects. We then made use of the HCP's 2 millimeter isotropic 3T resting state data to calculate the orientation dependence in tSNR. So tSNR is mathematically the inverse of the coefficient of variation that I presented previously. And yeah, here you can see nicely how those areas that tend to be more parallel in us have a lower tSNR, and areas that are more perpendicular have a higher tSNR. And so remember again, tSNR, it's kind of like currency of fMRI, tells you something about the ratio of signal fluctuation over its mean signal. And we evaluated this for smooth and non-smooth data, because I've been asked so many times, like, oh, can I smooth this away? Will it go away if I just, average over a big area? And so here we smoothed with a 5 millimeter kernel, which is fairly big, and doesn't help that much. so no. So this is now an orientation variability atlas. And what that means, orientation variability means where it's dark and kind of black. Those are areas where orientation is very diverse among us and where orientations average out. Whereas when it's light and bright, those are areas that are very similar among us when we lie in the scanner. And we can look at the impact of variability on tSNR variability. So here I'm plotting the variation of tSNR, sigma tSNR, as a function of orientation variability. And this sigma is a standard deviation over those 110 subjects. So that means how similar is the tSNR in FS average at this spot across the 110 subjects. And we can see that in the darker areas, which are more orientation diverse, the tSNR variability goes up. And in the bright areas, like somatomotor here, this is where tSNR is more similar across us. And tSNR often being interpreted as detectability somewhat suggests those areas might have a more homogeneous detectability and the dark areas have a more heterogeneous detectability. And we also note that the areas that have a higher orientation variability, which ultimately is a derivative of the geometric anatomical variability, is similar to the patterns of functional variability that are observed on the group level. So here, to the left, you have the variability atlas, and to the right is a map of functional connectivity variability. The blue areas are less variable in FC, so those areas, and are predominantly in the primary areas. Whereas the orange areas, orange and yellow here, are highly variable in FC and cover most parts of the association cortex, that is the higher order folds that are also responsible for higher order functions.
Olivia,
yes, please.
so were those analyses based only off of the surface normals, or did you also take into account head pose in the scanner? Since
yes. So for the analysis, yeah, it's always so what we do is we calculate the surface from the T1, and then we register the T1 to the EPI, and we basically take the surface and rotate it into the head position of the person during the fMRI scan. So the orientation is always calculated for exactly the orientation of the subject during the fMRI scan. Is that the question?
Yes, that's what I was curious about.
You have to.
If you had a particular region of interest that you wanted to focus on, I mean, this kind of implies that you want to load up your subjects in a particular way, perhaps with their head tilted a little bit. Or I guess you'll probably get to this. Do you think that there would be a way to correct for these effects?
So I don't think there's in that sense a way to, so you can't regress it out because it's not like motion that is adding to your signal, like a nuisance factor. It is a bias really in signal to noise ratio. But you can, I think, in that sense, correct for it that if you know that you have an area that is in an orientation that is producing a lower SNR, that you can, in your analysis, that you could put in a weighting for that. So I have personally not done any task data, but there is a nice paper by Alessio Fracasso who did task, I think it was a finger tapping task, where he showed how, I think the Z-stats was changing with, so he was looking at a piece of cortex that was highly folded and went through a bunch of different orientations. And he could show how, depending on the orientation, the Z-stats would follow the orientation effect. So if you're looking at one particular area, I think it would be a good cross-check to calculate the orientation for the ribbon you're kind of going along, and to see if you have a bias induced, and to maybe correct for that, or to at least interpret your data that way. You could think about like rotating your subject into a specific head position, but I guess that is tricky also in the sense the cortex is so folded. So I mean,
you would have to know in advance.
Yeah,
you
would have to know in advance, and yeah, it might be a little
the mirror
in practice. But yeah.
Yeah, thanks so much.
Yeah, no, good. Thank you for the question. So now I'm kind of like finished with all the gray matter stuff, and I want to dive into a little bit more of an exotic topic and look into orientation effects in both fMRI in the white matter. So here we see a nice rendered fiber tractogram from diffusion tensor imaging that I got from Zainab a couple of months ago for my ISMRM talk, and this is also how we linked up for this talk. And I really like this image because I can put it next to this image, which is a white matter microradiograph. And the microradiograph is an X-ray image where the vasculature has been injected with a radio-opaque fluid. And just by eye, we can see that vessels and fiber tracts are strikingly similar in their geometric path through the white matter in 3 areas. The most obvious is the radial path towards the deep structures and the brain center, but also at the sulcal banks. And both tend to enter the cortex at the gray-white surface in a perpendicular fashion. So there really seems to be some sort of vascular fiber coupling in geometry. So for the remainder of this talk, we will hover into the fibre-vascular coupling in the deep white matter, and this observation raises the question if vessels are simply packed between fiber bundles to optimize space, or if vessel geometry actually reflects blood flow regulation that might be linked to physiological or neuronal activity going down these vessels. So we used the geometric coupling between peel surface vessels and the cortical surface previously to infer the peel vessel orientation. And here we will use fiber directions or fiber direction estimates to infer vessel orientation to the B0 field axis. As we discussed before, BOLD strongly depends on vessel orientation to B0, and we will use this dependence to identify dynamic sources of BOLD in the white matter. The motivation behind this analysis is the topic of white matter BOLD fMRI, which is a topic that is strongly debated in the community. And already 15 years ago, the first BOLD signals were reported in the corpus callosum with interhemispheric transfer tasks. And the number of reports is increasing, and many results might be relatable to fibre-vascular coupling. I would like to point out to this study by the Gore group that derived fiber tractograms from spatial correlations in resting state fMRI data. So this thing here was entirely produced from fMRI data, no diffusion imaging. Systemic BOLD effects in the white matter could, for example, arise from respiration-induced changes in oxygenation. We would suspect these changes to be global, so we would suspect that they would present in the entire white matter. But local functional hyperemia, as we observe in the gray matter with neuronal activity, is questionable. White matter metabolism is not oxidative, as is gray matter metabolism. And also remember that synapse density is about 80% lower in the gray matter. So it's okay to be cautious about white matter BOLD, I think. So here we test for dynamic sources of BOLD as expected from venous vessels along specific fibers using orientation effects. And let me explain how we do this. So we saw previously that the static gradient echo signal in a voxel depends on fiber orientation to the B0 field. We know this from the literature. This has been nicely shown in R2 star and T2 star value dependencies with cosine squared and sine squared. And the same will hold for a static vessel that runs parallel to a fiber and doesn't undergo any changes in volume, flow, or oxygenation. So let me explain this with a simplified voxel cartoon model of cylindric fibers and vessels that run in parallel. So if parallel to B0, myelin and blood will induce no susceptibility field offset in their surrounding, and the gradient echo signal will be maximized. But if the structures are perpendicular to the surrounding field offsets, the field offset are perpendicular to the V0 field, the field offsets will be maximized and there will be maximum signal dephasing and your gradient echo signal will be minimal. So this is the kind of like static signal that we would expect in gradient echo. And that would also be what you would expect if you just run a bold EPI scan in the white matter and look at the mean signal of your voxel. When it comes to dynamic signal variations over time, the static structure should induce an amplitude fluctuation that is proportional to the static signal and follow the same orientation signature. So if we assume we have a voxel that cover like myelin and vessels and the subject is moving and we get a fluctuation in the signal, we would expect that to run in the same manner. However, dynamic changes in bold in the vessel itself should exhibit the opposite negative orientation signature, here shown in purple. And that is because when the vessel is parallel, remember, there's no extravascular field offset. So if anything in the vessel changes in terms of blood volume or oxygenation, that will not be detectable because the extravascular field offset can't change. Whereas when it's perpendicular, the extravascular field offset is maximized. So any bold change from the vessel will be detectable in these extravascular field offsets. And this is really what I want you to remember from this, because this is a little bit complicated and complex. Dynamic bold from vessels should have an opposite orientation signature to static components. That's all you should remember. To do this study, we took the 110 subjects that I showed earlier for the orientation atlas. It's from the Human Connectome project, which provides spin echo diffusion data. This data was processed and prepared by my colleague, Qian Tian. We estimated fiber directions using the deterministic tensor fit to the B1000 data, and we repeated also the entire analysis with probabilistic bedpost extractography that gives us 3 direction estimates. So here I would like to point out that most fibers love to cross and kiss. That is very well known in the literature, but vessels are tree-like structures. And to my knowledge, no one has ever observed crossing vessels in the brain. Sometimes vessels coil, but this kind of like crossing that fibers do, that's really something that vessels really don't really do. So fiber directions are then rotated into the head position during the fMRI scan to calculate B0 orientations during the resting state acquisition. And we use an MNI-based atlas of fiber density from the IIT Human Brain Atlas to create fiber-specific masks. And the fMRI data is the standard gradient echo API. And we take the time series temporal mean as the static signal and the temporal standard deviation as the dynamic component sigma. So these are the results of fiber orientation dependence of the static fMRI signal in the global white matter. The orientation bias is 11%, which translates to a change in delta R2 star of 3 Hertz. And this is also kind of like what goes in line with literature values that you find on white matter GRE dependence when you rotate the white matter and calculate the orientation of fiber to B0. Then in the next step, we calculate we plot the dynamic fluctuation of the white matter time series over the entire brain. And what we find here is that this dynamic component, so the bold fluctuation, has this funky biphasic behavior that is really inconclusive within the error bars. So I wouldn't say that there is any interpretable orientation effect in there. So let's check the results in the individual fiber bundles instead. Because remember, this is really pulling from the whole white matter. So here's a Giphy run through of all fiber bundles. We can see that orientation dependencies within the individual bundles are diverse, and the dynamic fMRI fluctuation has distinct orientation signatures in different bundles. Many are showing the negative orientation bias of dynamic bold, but some don't show this at all. So first of all, you should notice that within all of these fibers, all of them show the same positive orientation effect in the static signal. So everywhere we suspect that we have myelinated fibers and potentially vessels that follow this orientation curve. But then we can see how here, for example, corpus callosum has this negative orientation bias, where previously the cortical spinal tract shows the other way around. And we ranked this now in numbers. So here we rank bundles by their orientation signature and overall bias magnitude from highest to lowest. The highest dynamic bold orientation effects are observed in the inferior fasciculus, the corpus callosum, and the optic radiation. Those are also the fiber bundles that have been pointed out in bold fMRI studies throughout the decade. We also find bundles that don't show dynamic orientation signatures, like the cortical spinal tract and some fibers that are a bit inconclusive. To validate our findings further, I teamed up with my colleague Mike Bernier, who has developed the segmentation tools to segment and reconstruct whole brain vasculature from ferromoxidil enhanced multigradient echo scans. This contrast agent allows them to enhance vessel detectability. So here you can really nicely see this is the pre-contrast scan, this is post-contrast scan, like the amount of vessels that you can see, it's wild. And in particular in the white matter, where vascular density is really lower than in the gray matter, this really nicely boosts what we can detect. And also remember the microradiograph that I showed you before, this is really closer to it. And he's creating these really useful vascular density maps from it. And he created bundle-specific vascular density maps, as you can see here. So here for the occipitopontine tract, inferior fasciculus, and longitudinal fasciculus. So these color codes, those are the fiber tracts, and this kind of like golden thing here, this is the segmentation of the vascular density. So here's a comparison of our results. So we threw together our data. Mike ranked the top 10 vascularized fiber bundles from highest to lowest. Those are the orange-green dots that you can see here. In the middle, the red dots are the orientation bias in percent in the static fMRI signal from my data analysis. So this suggests that the observed orientation dependence is likely not only due to myelin, but might be further enhanced by the amount of vessels that run in parallel to these fibers. Similarly, the purple dots to the very right here show the dynamic fluctuation bias from orientation. And also here we match up pretty well in terms of the more vascularized a fiber bundle, the more it displays this dynamic bold related orientation effect that would not be explainable by myelin structures. I would also like to point out here that we are really comparing different data sets here. So my data set, this ranking, that's HCP data. Mike's data set, the vascular density, is coming from 5 subjects that he scanned on our 7T scanner, and we're just comparing our results in MNI space in these bundles. So that is kind of encouraging that what we see here might be really pointing towards a meaningful direction. Last but not least, I would like to show you the bed post-X results because I think it's very interesting. So here you can see the same Giphy run through as before, but now for all 3 direction estimates. And we see that the most dominant direction, this is the darkest spot, tends to show the highest orientation effect. But let's discuss some of these cases in more detail on the next slides. So for common resolution, we know that most white matter voxels contain definitely more than 1 fiber direction estimate. And here's an example of the frontal ascending tract. We see static orientation bias among all 3 direction estimates. And all 3 directions also show a dynamic orientation signature that scales with the static orientation component. And we suspect, because the orientation bias is positive and fairly slow, that maybe the frontal ascending tract it's just not very much vascularized or not vascularized at all. And also going back to Mike's ranking, the frontal ascending tract didn't even make it into his top 10 fibers that are vascularized. Here's the corpus callosum, which is known to be more unidirectional. And we see that the dominant fiber direction shows a dynamic orientation signature associated with dynamic fold changes. The other estimates show little orientation effects just because maybe the other directions are not really present, or also do not carry any vessels. The paritopontine tract is interesting. Here it is only the minor direction estimate that shows an orientation effect from dynamic fold, and maybe it is only a small, only the minor direction that has vessels running in parallel. But we also see examples like the optic radiation where multiple directions are present, and all of these directions carry dynamic orientation signatures. As mentioned before, we do not expect crossing vessels, and it would be interesting to see if this is really just a resolution effect and if it would dilute away if we go to increasing resolution and where we would sample fewer fiber and vessel directions in the voxel. I would like to conclude this part with 3 take-home messages. First, the fMRI signal in the white matter depends on fiber orientation. In the HCP protocol, this is 10%. Dynamic orientation effects are heterogeneous and more prevalent in specific bundles, such as the inferior fasciculars, corpus callosum, and optic radiation. This suggests that there might be dynamic ball changes in the white matter. It is not clear, though, if this is bundle-specific signaling activity or if the origin is a more global systemic physiological change that presents in downstream white matter vasculature. The result should also raise caution for regression methods like COMCORE, where white matter RIs are used as nuisance regressors. So I want to finish this talk with an outlook and a bigger picture on anatomic functional coupling, and also tie this into the work that our lab is doing, because we are all kind of pulling on the same rope for the bigger picture. So our lab has a strong focus on high field, high resolution fMRI. My colleague Jörg van Müller is taking this powerful van simulation approach to the next level, and he creates the so far most realistic simulation of hemodynamic responses from first principles. And I hope I convinced you how powerful these vans are. So Louis was really able to predict these orientation effects using the van model from first principles. So I think this kind of modeling is a space to watch, because it really allows us to use realistic vascular networks to really get a realistic fMRI response. As I mentioned early on, the orientation effects were already mentioned by Ogawa, but in the early days, resolution was too coarse to make people worried. But the methods are becoming available, and my colleague Avery Berman is pushing limits with his variable flip angle fleet acquisition. Here, he just published this. He is running 0.6 millimeter isotropic functional imaging at 70 here. That's for a visual stimulus. He also works on higher resolution spin echo EPI here at 3T. Oh, sorry, here at 3T, which is particularly interesting as it is less vulnerable to the Peel vessel field offsets and more specific to the microvasculature. So that's, I think, also an exciting technical development. And last but not least, we are working on quantifying the whole brain vasculature using paramoxyl enhanced angiography. And we're just amazed by the amount of vascular structures that exist. The gray matter vasculature is fairly studied, but the white matter vasculature, I think there's still so much that we don't know, but that really is worth studying. I think there's a lot of interesting things that we can learn in the upcoming years from these new technical developments. And to even tie this work into a bigger picture, I hope you learned something about particular orientation effects and the importance of knowing the vascular anatomy to interpret bold fMRI signals. But there are many more anatomical, subject-specific, but also hardware-specific sources of bias that ultimately reduce reliability in the single subject and the group scan. So I want to mention cortical thickness for an example that varies non-uniformly across the brain. For example, in your visual area, the cortical thickness is like very thin, and then it gets thicker for the temporal lobes. And that will induce partial volume effects, which will be more pronounced in the thinner parts of the cortex. And there, so here in this plot, you can nicely see how your z-stats will depend on the partial voluming. So z-stat variability will depend on cortical thickness variability. And bear in mind that cortical thinning is one of the most prevalent observations in aging and neurodegenerative diseases. And a lot of studies use fMRI to infer neuronal changes in aging and neurodegenerative diseases. So learning more about functional anatomic coupling and its impact on the fMRI signal, I think, is important, just to make sure we interpret our results in the right manner. And the last bias, and then I will be finished. The last bias I would like to put on your radar is a hardware bias, because people might change their head orientation in the scanner, and people have different head sizes, etc. But the coil is always fixed. The coil is always fixed in size. So coil sensitivity will vary across the cortex in terms of like the distance from the area to the next received element, but also with the variation of different head sizes. So if you have someone who has a smaller head, then the distance from front to the next received element will vary on that. And you can see this in these maps. So here, this yellow map, that is the mean received sensitivity of the 70 coil that is provided by Nova Medical. You can see you get high sensitivity in the periphery, but you get this very low sensitivity on the bottom. That's the famous Nova Medical drop-off for those that do 7T scanning, as it said. But here, this is a variability atlas of received sensitivity of the Siemens 3T head coil. And you can see that the frontal part is more variable in sensitivity, and the back, because those are depends on padding and maybe also thickness of your skin and skull. And yeah, as I mentioned, the frontal part has this varying distance to the received elements. And with this, I would like to thank you for your attention. And I want to thank everyone at the Martino Center who helped me to create this work, in particular my boss, Jonathan Polimeni, is very resourceful, and Kirian Tian and Michael Bernier, who were particularly involved in these two projects that I presented. And yeah, shoot away with questions.
Thank you so much, Olivia. All right. Feel free to unmute yourself and ask a question, or you can chat it to me.
I have a question. So you were talking about how there are some areas of the brain, if I understand right, that have more sort of homogeneous vasculature in addition to the white matter or the gray matter, and other areas that tend to be more heterogeneous in their vasculature, correct?
So what I was talking about, heterogeneity, I didn't mean the vasculature being more heterogeneous, but the cortical folding pattern and the anatomy. So the variation of the geometry of the vasculature with respect to a common axis, in this case, the B0 field axis. So if you have an area that will vary more in geometry across us, that will mean that the vasculature will also change its position to B0 more across us. And vascular heterogeneity, I'm sure that this will exist as well. So I'm sure you will get areas that have a more similar vascular density across us than others, but I'm not an expert in that, or I'm also not sure if this has been properly investigated.
Yeah, I wonder with the work that you're doing if it may at some point lead to that, because in addition to changes in the dynamic bold signal, which you were talking about as well, I wonder if there will be a return to sort of mapping differences in vasculature and the way that actually affects the bold signal, but the actual neuronal activity there. Because I wonder if there is a difference in, if there are regions that have more heterogeneity or homogeneity of vasculature, if that has an effect on actual functional, the functionality and the functional activity of.
Yeah, we ask ourselves kind of exactly the same question all the time. It's a bit of chicken and egg problem.
Yeah.
Because the question is what was there first, functional variability, and then that comes with anatomical variability, or was there first anatomical variability and that drives functional variability? Also, this atlas that I showed, comparing with the Buckner results on functional connectivity variability. Mm-hmm. I don't want to claim that orientation variability or geometric variability produces functional variability. It might be something that is very tightly coupled. So maybe if you have an area that has lower neuronal activity, it doesn't need so much vasculature, is less vascularized, or an area that, maybe that gets trained, maybe it will develop more vasculature over time. Those things I think will really tightly go together, and that the same with cortical thickness and vascular density in different layers. I think we will have to study them in parallel.
Yeah, that's fascinating. Thank you.
Yeah.
Do you think that these kinds of effects could be corrected with, you know, individual subject angiography, like with ASL or something like that?
So if you have a CBV-based fMRI method that is not relying on susceptibility effects, so, you know, like something like VASO, for example, or ASL, should not be subject to the orientation effect. I mean, depending on what your readout is, if you use an EPI readout, you know, you get your T2 star again.
Oh, that's actually a good point, though, that I could see that. But I guess what I'm getting at is that if you had, you know, an angiograph for one subject, for example, and computed, you know, vessel orientation at a voxel-wise basis, you know, you could potentially use that maybe as a regressor.
Yeah. So so the idea is a little bit, the more we know about the vasculature, the more we can maybe go towards like an informed fMRI analysis and throw that in, like similar how you could maybe use partial volume maps to throw that into your analysis. I think it will, it won't be that you can regress it out, because as I said before, it's not like motion or breathing that adds to your signal. It's not a time varying factor. Right. It's really biasing your signal to noise, your contrast ratio in that voxel. But in your statistical analysis, you might be able to use that term to, you know, boost up those areas that are suffering from like a disadvantage orientation bias.
Yeah, I was more thinking for a, you know, a second level analysis. So, you know, for a given voxel across subjects, maybe some of them have, you know, vasculature that's parallel with the B0 orientation, and others are not. So that might be able to deal with variance associated with that.
Yeah, yeah. So yeah, I think this is something that you might be able to do. I think ultimately getting like a good vascular, like a high resolution vascular angiogram that resolves the peel vessels is like really, really hard. So I think it's not super realistic, or at least not within the next couple of years, maybe 5 years down. But I'm wondering to what degree we will also get a lot of help from what Mike is doing, who is really working on atlases of vascular density, which will be maybe helpful on the group level, that you can throw that stuff into your analysis.
Thanks.
So I have a question. So basically, is it correct to understand this, like where the vessel is perpendicular to the B0 field, it gets more signal or stronger signal?
It's the other way around. If the vessel is parallel to B0, this is when the intravascular field offsets are maximum, but the extravascular are 0. So I can maybe, hang on, I'm just gonna go back to one of these slides. Maybe. Okay. Go to the very first one. Yes. So here the vessel is parallel, and the extravascular field offset is 0. The intravascular is maximized. Can I play this in here? Yeah. And stop. Okay, well, almost. Now it is almost perpendicular, and the extravascular field offset is now maximized, and the intravascular will go to like the opposite direction. And it is the extravascular field offset that we mainly care for, because that is covering the most of this space where we're sampling from, and which is also getting dominant, like really dominant above 4 Tesla. If you go too low, if you were you acquiring data at like 1.5 tSNR, I'd actually be more like, I'd expect the orientation signature to reverse to actually follow the intravascular one, because extravascular field offsets at lower field like 1.5 are definitely lower than the intravascular one.
So,
But in 3T70, rule of thumb, vessel parallel, not very detectable, vessel perpendicular, signal more detectable.
so if that is related to the cortical folding, and we know that is developed from the neonates and to the adults. So do you think the differences we observed between group can be due to this, the orientation differences?
I don't think that we can explain complete group differences between developing children and adults just based on the anatomical change. I think it definitely has an impact on that. And again, what the question that was asked earlier, it will be the question, like how much are they showing kind of the same thing? Like there will be, I'm sure there will be change in like functional properties from growing up to being an adult, but that goes in line with anatomy and how much are they acting against each other or with each other. And I think one thing also to bear in mind is in terms of the homogeneity, if we look at tSNR variability, you will just get certain areas that will be harder to detect on the group level if they vary more in anatomy across us. Whereas areas like somatomotor that are geometrically, anatomically more similar across us, we'd expect to get more results, better results on the group level just because every subject will be more similar. But yeah, I wouldn't put myself so far out that I say that geometry will explain all of the differences that we see between groups.
Thanks.
Any other questions? I just have one short question, and I don't know if this has been explored, but do you know about the effects of what the vasculature looks like in neonates and within the first 2 years of life?
No, I don't. We are generally, I mean, we are looking so much to get any information about vascular and vascular changes with development, and we already struggle to get good information for white matter. Like no one, we haven't found a person yet that knows of any good literature on like how the capillary network in the white matter looks like. Mm-hmm. And if you look at newborn infants, I haven't seen anything particular, and I think it's also hard to study because if you look at all of the studies on the cerebral vasculature, they're usually very old brains of people that died, you know, when they were like 70 or 80. I don't think there are post-mortem studies of newborns that really look at the vasculature. But I'll put it on my list. I'll ask around a little bit and also ask John if he knows of anything. That's a really good question.
Yeah, I mean, especially if that mirrors the your findings about where the greatest variability is, I think that would be informative too. Okay. Well, thank you so much again. And any other questions that you guys have, you can feel free to email Dr. Viessmann. And thanks again for joining us, and have a great Friday, everyone.
Thank you. Bye, guys.